I'm Nadeem Silverman.
I'm Alexandra Griffin. We go to Bard High School Early College in Manhattan. Our project was the effects of seawater flooding on microbial biodiversity in East River Park.
We worked 7 hours straight, barcoding DNA, extracting DNA, and throughout that 7 hours, we didn't even know if we had DNA. And in the very end, we were able to see if all of our hard work paid off, and it was. I've never felt more satisfied when I just saw that there was something in our sample and that we were going to be able to continue and not do another day of that kind of work. So I have more respect for the work scientists are doing every day.
Science requires such a level of meticulousness that I didn't really expect. It's not all sudden breakthrough, I get it now. And so kind of that you really need a kind of patience and tenderness with kind of whatever you do, and that sort of tenderness was not something I had associated with science before doing this.
We were able to mark certain growth patterns, and that after events like Sandy and just general flooding, you do see regrowth in certain populations.
For me, one of the most interesting parts of this project is kind of the urban planning aspect of it and thinking about how to create more sustainable and resilient cities, and that's something I'm really interested in and might be something I go into in my future.
I'm Andre Latun-Basoon, and the name of my project is Discovering the Origin of the Introduced Malagasy Giant Chameleon in Florida. My favorite part was actually cleaning the sequences on the computer, because you get to see the exact similarities and differences and get to edit and cut out and paste and stuff. So I thought that was cool, and the fact that the lab work was way different than anything I've ever done. I don't know, I just liked how meticulous it was. It was good.
We're both from the Queens High School of Teaching, Liberal Arts, and the Sciences, and our project's name is The Human Microbiome. Microorganisms are present at different intervals of life and death. I personally enjoy running a gel electrophoresis. I think it's the funnest part of being in a laboratory, just working with gels, agarose, and pipettes. It's so much fun.
The science is a lot more hands-on. Basically in school we just do notes and little labs that are really simple, but this is really fun. You get to learn a lot more vocabulary and you get to learn more about everything in detail.
My name is Larry Chan and I'm from High School for Health Sciences and Human Services. Basically what we did was we went around local supermarkets and we went to collect fish labeled as red snappers. Well, we found that there was not a lot of real red snappers. It introduces you to lab work, something that a lot of schools, especially public schools, don't do a lot. Not as well as I would like.
My name is Felicia Howe. I go to Stuyvesant High School.
I'm Carrie Yim, and I go to New Explorations into Science, Technology, and Math. We found that there was no genetic variation between the farmers market eggs and the supermarket eggs. I think the most enjoyable part was working in the lab, because in school labs we don't get to work in such high-tech equipment, so working in the museum lab was really great.
So it was really great to be able to work with a scientist from the American Museum of Natural History. She's just a really great scientist, and we were able to learn a lot of things about laboratory technique.
My name is Brian Suarez. I go to Benjamin N. Cardozo. I am a senior, and the title of my project is a validation study of bacterial species in probiotic food products. Probiotics that are being advertised in yogurts, not all of them contain that probiotic. Typically, what we found was it's bacterium commonly found in bird feces. Most enjoyable part? Probably, growing the plates. It was actually a lot of fun because I got to spend a lot of time with my mentors, and they're actually very supportive of my project. So I think I have to thank them a lot. I've always known that I wanted to do research, or I wanted to go into the medical field, but this project really helped me further validate that opinion, because I love research so much. It really brings me passion and joy.
My favorite part of the project was actually analyzing the samples. When finally coming to the realization what the identities were a blessing to me.
It was very fun creating the TAC master mix for the polymer machine reaction, PCR, and just gels. It was very fascinating to see them under UV light and understand how these, the ladder, the base pairs work. We actually did over 1,000 reference sequences for a bold database when we identified the sequences of our 50 samples.
We tested fast food meats and grocery store meats to see which one had a higher rate of food fraud.
For the grocery meats, everything was what it said it was. That's very good news. Well, for me, the hardest part was actually trimming the sequences that we had. That was a lot of work, and we had to decide which ones to delete.
The best part was probably just conducting my own research because this is my very first experiment and I'm really excited about it. And I got to have a really nice mentor who worked with me throughout the whole project. It was really cool because I never worked with a scientist before, and it gave me a lot of experience.
It was a great experience. Like, you never have that one-on-one experience in a classroom with 30 students, especially with a certified like scientist with her PhD and everything that she does. This is her field of work, what she does every day, and it was a great experience.
We've always done a smaller scale laboratories, but we've really never been at this grand scale where we present this and we make such huge posters. It's never been like this. It's just a small lab, one period, and that's it. But nothing compares to the experience and the thrill that we got from presenting here today.
Well, the most challenging part was actually creating the poster and writing out all the things.
Well, my most enjoyable part was doing the DNA extraction, and that was really fun for me. I've never done anything like this. Like, this is the most fun project I've ever done in my life, and I really enjoy it, and I'm proud of it.